Yes. Is this working? Okay. That tapeworm really did come out of that small fish, about a 3-centimeter long fish. In fact, it's not even a particularly big example of this tapeworm. I found 3-spined stickleback, that's this fish species here. Some of them can be as much as 50% tapeworm by mass with a single parasite. Now, I imagine half of you are thinking, thank goodness people don't get that kind of parasite. And maybe the other half are thinking, thank goodness we didn't talk about this right before dinner last night. But my initial reaction in seeing this particular picture is to say, cool, look at that. And by that, I don't mean the really big parasite. I mean the really small one. These two parasites are the same species. They're the same age. They're 42 days post-infection in a lab experiment. And they're full siblings. They're sisters. So if they're the same age and the same genotype, more or less, why is one massive and the other minuscule? The answer, it turns out, has to do with the fish from which they were removed, respectively. Same species of fish, just from two different lakes a few kilometers apart. One of these populations, actually quite a few populations, have evolved an immunologically novel strategy that lets them suppress parasite growth dramatically. I'm interested in understanding how they do that from a genetic and immunological standpoint, but also why some populations do this and others do not. What's the difference? Now, to be fair, people don't get that parasite. You could actually, if you ate a raw stickleback, which is a very bony, spiny animal, you wouldn't want to do it. But we do get our fair share of parasites. Estimates range from about 1/4 to as high as 1/3 of humanity are affected by macroparasites, helminths, tapeworms, nematodes, and their ilk. These present a massive public health burden, a massive economic burden. They strain health systems, especially in developing countries. And they're so effective and such a big problem because they have evolved strategies to avoid our immune system. They fly under the radar or they jam our immune system for their own benefits. They can establish infections that last months, years, a lifetime. And what we know about controlling these organisms relies very heavily on our understanding of our immune system. Our bodies have a complex set of organs and tissues and cells that search for invasions of this kind and try and eliminate them through really mind-boggling arrays of monitors and response systems. And much of what we know about this immune system comes from really phenomenal body of work with lab mice. And lab mice and a number of other model organisms like zebrafish and xenopus and so on are so effective because we can manipulate them in the lab with incredible precision. And we can do that because there are tools and technologies and reagents. You can order a single genotype of mouse again and again. You're working with the same organism. You can order gene knockout mice. You can order monoclonal antibodies. It will label proteins. You can track the immune kinetics through time, spatially in a tissue. And that's incredibly powerful. But I'll argue that there are some limitations here. And one of the core limitations is that this cage full of wood chips is not where that immune system evolved to work. And that matters because our immune system is hugely context dependent. So instead, my lab works in places like this. This is my laboratory. I have one at the University of Texas as well. But this is our mobile immunology lab. It's a trailer. It's an RV. And we have a flow cytometer and cell culture incubator and so on. And we can bring that to the side of a lake and actually assay immune function in animals fresh out of the environment in which their immune system evolved to act and currently acts. Now, let's be clear. Studying immunity in a non-model organism in the wild has all sorts of challenges to it, right? Because we're dealing with an uncontrolled experimental system sometimes. We do lab work as well, as I'll get to in a minute, but we're dealing with a lot of heterogeneity, environmental heterogeneity, genetic diversity in the wild that poses a challenge. And we don't have all of those monoclonal antibodies and knockout isogenic strains that make mice such a powerful tool. So we have our limitations, but we have some benefits as well. And one of the benefits is that these are a really neat animal that show phenomenal variation among individuals, among populations. This is a 3-spine stickleback male here, looking a little confused after a female just dissed him. This is a great model organism because of its diversity. It exhibits heterogeneity among individuals and from one lake to the next, from lakes to streams to ocean sites, and it's found in a large number of populations around north temperate environments in the Pacific and the Atlantic. It's abundant, it's easy to catch, it's easy to manipulate in the wild and in the laboratory, and it's host to this lovely animal that I've already introduced you to, Schistocephalus solidus. This is one of the oldest known examples of a complex life cycle parasite. Its eggs hatch in fresh water and only in fresh water, which will matter in a moment, and it infects those copepods, those little zooplankton, which are eaten by a variety of fish, but only in this species' 3-spine stickleback will that parasite grow to its full size in the body cavity, kind of worming its way around the organs. And when it reaches that massive size that you've already seen, it helps the fish get eaten by birds. It makes them more visible, slower. It makes them actually gravitate towards the surface where things like loons can catch them, and the parasite breeds in the loon's intestines. Eggs are dumped into the water again. Now, I don't need loons in the lab, which is good. We can trick the parasite into thinking it's in a bird just by putting it in a nice warm, dark water bath, and it'll mate for us, which means we can do genetics on both the host and the parasite concurrently and pursue what I call reciprocal genetics, which is the mapping of the interaction between two different species genomes. Another great feature of this system is its natural diversity that I've already alluded to. The 3 fish shown here are all male stickleback from 3 different lakes, and their color variation and their body shape and structural variation and physiological variation evolved very recently, as I'll get to in a minute, and provides a target for evaluating the basis of adaptation. The same is true for the parasites, which adapt in turn to deal with their heterogeneous hosts. Now, to be fair, a lot of immunologists have worked very hard to eliminate genetic diversity in lab model systems, and that's for good reason. What we'd really like to do is take a bunch of identical genotype animals, treat half of them in one way and half of them as controls, and look at the difference, and you know that your manipulation caused some phenotypic effect because there's nothing else varying. Naturally heterogeneous organisms, you can't do that because there's so much variability, but it has its benefits. So a lot of immunology has been really resting on work involving forward genetics, and Bruce Beutler received his Nobel Prize for this kind of work. And the idea is you take a relatively homogeneous population of lab animals and you mutagenize them either randomly or in a directed manner, and you ask, does changing gene X have a consequence on trait Y? Does the mouse lose immunity to some parasite, get sick, die, et cetera? And then you know very clearly that that gene you changed has an effect on the phenotype that you're observing. The problem with this approach, and it's very powerful, I don't want to critique it, but its one limitation is that anytime we're knocking out genes like this, we're typically creating deleterious variants. Right? This is a loss of function, usually. And what I want to know is how can we improve upon an existing immune system? What can we do for value added? And instead of using forward genetics, we use what's called ecological genetics, which takes advantage of natural selection as a natural genetic screen. And the idea here is a single lake on Vancouver Island might have 1 million or 500,000 stickleback in it. And imagine some small fraction have a variant that is protected against this parasite. Maybe 1%. It might be a new mutation, might be immigrants from elsewhere. Now, the other fish, some of them may get this parasite and either get eaten by birds or fail to reproduce, and their alleles are removed from the system. And in a single generation, we've just doubled the relative frequency of the protected variant. You do this a few generations in a row, and your population is entirely protected. Now, selection is scanning 1 million fish per lake per year for a protected variant. Repeat that for 10,000 years on 10,000 different lakes, and you are going to scan billions upon billions of animals for beneficial novel variants. For example, on Vancouver Island, where I do a lot of my field work, the island was under a mile of ice just 15,000 years ago during the Pleistocene glaciation. So there were no stickleback there. There were no lakes, there were no streams, and instead the fish lived in the ocean along the shoreline. And when those glaciers melted, those fish in the ocean had access to new environments, newly available lakes and streams, and they moved from the ocean into fresh water. And importantly, they did so in a replicated way. Every watershed, every river basin that feeds into the ocean was separately, independently colonized by a single pool of oceanic fish that persists to today largely unchanged. Which means that we can sample the ancestral type, the marine fish today, and we can sample the derived freshwater types, and they are vastly different. Those freshwater fish have, in 12,000 years, evolved numerous adaptations to fresh water because they encounter new food types, new predators, and new parasites. For example, on the top, that little copepod there is the food source by which the fish get the tapeworm I've told you about. Sure enough, only in fresh water do these fish get an appreciable number of these tapeworms, and only in some lakes where the fish are encountering this food item on a regular basis. The marine fish really almost never get this parasite. And we had to wonder, is that because they're immune or because they don't encounter it? And the answer is it's because they don't encounter it. It's an ecological control. In laboratory infection trials, those oceanic fish, 90% of them get infected. They're super easy to infect. But the freshwater fish, only 12,000 years derived from those marine ones, about 10%, 20% infection rate. Not only that, but it's evolved repeatedly. In Canada and in Europe, we've separately confirmed that newly evolved freshwater populations have evolved this resistance. How do they do that? Well, we've made the most progress so far in answering that question actually by focusing on two freshwater populations, Roberts Lake on top there and Gosling Lake on the bottom in the pictures. Gosling Lake typically has more than half of the fish are carrying around that massive parasite. More than half of the animals. On the other hand, Roberts Lake, a few kilometers away, I have never seen this parasite in more than a decade working there, even though they're eating the same food item. And the loons are there. Everything is in place for this life cycle of the parasite to persist, and yet those fish do not get the parasite. So we wondered, could those Roberts Lake fish be resistant? Bring them into the lab, breed them for a couple of generations to remove environmental differences, epigenetic effects, infect them experimentally, and we get 20% infection rate in both populations. So it's not an infection success in terms of initial establishment. Instead, it's parasite size. The worms on the left and right are full siblings. The difference is the fish from which they were removed, and you can see some data on the side there. We have Roberts Lake genotypes and Gosling Lake genotypes on the edges, and various hybrid genotypes in between. We have additive genetic control by the host genome of parasite growth rate over 2 orders of magnitude. So it's about a 100-fold difference in parasite size. How do they do that? Well, it turns out every single immune phenotype we're able to measure in this non-model system differs between these fish. Every phenotype I've ever measured. So it's hard to pinpoint exactly what's going on just by a quick assay. So then we did some genetic mapping, and the principle here is you take pure susceptible and pure resistant fish, you hybridize them. F1 hybrids have half of their genome from one, half of their genome from the other. When you mate those hybrids, their genomes break up and get shuffled by recombination, and we end up with a large pool of F2 hybrids and back crosses. We've experimentally infected nearly 1,000 of those, and what you then do is you genotype markers across their genome, we experimentally infect them, and we ask, is there a difference between the fish that get big tapeworms and small tapeworms? For example, in this window right here, if you have a small tapeworm, you have entirely red ancestry and blue ancestry in the big tapeworm fish, respectively, in this cartoon. Well, here's a genetic marker on chromosome 12 that is strongly associated with differences in parasite mass. We have found 5 such markers in the stickleback genome, 5 genomic locations that are powerfully involved in regulating parasite growth rate. 5 genes. What are they? Well, we've done transcriptomics to map changes in gene expression between these populations. And this is, for example, one transcriptomic analysis. Every dot there is a gene transcript. And the y-axis is the magnitude of upregulation of a gene in a context-dependent manner. That is to say, in only one genotype, only after infection by the parasite. So what are those genes, and particularly that focal gene there, that is massively upregulated? Mostly the genes that we're finding are involved in fibrosis, which I'll explain in a moment, and reactive oxygen production. And those genes fall within those markers in the genome to which we have mapped parasite growth rate. So sure enough, fibrosis seems to be the most interesting phenomenon. This is a whole body cavity scarring formation. All of the organs are bound together in this tight fibrous mass that captures a parasite and holds it fixed and typically ends up encasing it in a cyst and killing it. So in the long run, those tiny tapeworms can be eliminated successfully. So these fish are successfully capturing and controlling the tapeworm, and in the wild, this happens as well. So this is a survey of 10 populations. Every dot here is a different lake, and in lakes where every infected fish has fibrosis, their tapeworms are tiny, microscopic. And in lakes where the fish do not get fibrosis, their tapeworms are massive. And so both in the laboratory and in the wild, we have evidence now through multiple sources that fibrosis genes are involved in suppression of tapeworm growth. We established that furthermore by a genetic mapping study across many populations. We sampled 68 different lakes on Vancouver Island. We sequenced over 1,000 fish from more than 10,000 genetic markers and asked, are there any genes that predict individual fish's parasite size and presence and absence? Those genes are marked up here with high association with parasite infection status. Of the 20 most strongly associated genes, 13 of them are immunological, and half of them are involved in fibrosis or reactive oxygen. So we're pretty sure we have candidate genes that we're involved currently in trying to knock out, to manipulate experimentally in the lab for confirmation. So the punchline here is that we have rapid evolution in 12,000 years in multiple independently evolved populations of regulatory changes in fibrosis that lead to this massive whole body fibrotic response that suppress parasite growth and represents an adaptive response to protect the fish. There's a catch. Fibrosis is, of course, not good in other regards. Namely, female fish who get this fibrosis response do not reproduce easily at all because that fibrous mass prevents ovaries from growing and maturing, 80% loss of reproductive capacity. Males, on the other hand, suffer no penalty at all for the fibrosis. Females bear the burden of this immunological protection. That's fascinating because humans, of course, have a large number of diseases that depend on fibrosis and are involved in fibrosis. So we have identified now, therefore, a naturally evolved, adaptive, beneficial fibrosis susceptibility. And we're hot on the heels of what genes and particular pathways are involved in turning on this pathological susceptibility to fibrosis, because in the cost-benefit balance of things, it's better to not get that terrible parasite than it is to have fibrosis. Turns out the parasites have something to say about this as well. We're also doing genetic mapping of the parasite to find out how some genotypes of the parasite shut down fibrosis in their host. So the punchline here is that by studying ecoimmunology in the wild, function of the vertebrate immunity in a natural setting where there's genetic diversity in the host and the parasite, there are many parasite species acting concurrently, there are many sources of stress. These fish don't just worry about parasites, they worry about finding a meal, finding a mate, not getting eaten by a bird or a trout. And those multiple sources of stress have effects on the immune system that are not effectively replicated in a laboratory setting. And so the final thought that I want to leave you with today is, how well do our lab models really replicate immune function in natural settings where immune function evolved to work and works today. And a variety of lines of work in my lab suggest that some aspects of immune function we can, in fact, study in the lab pretty effectively. We found fibrosis in the lab, and that brought us back out to study fibrosis in nature. But other phenotypes we cannot replicate in the lab, yet we observe in nature, or vice versa. And I think that's something that's really troubling in some regard. And I argue that it means that we shouldn't throw either baby out with the bathwater, but we need it as an integrated system to study immunity in both a finely controlled setting and out in nature. And with that, I need to thank a number of people, too many to put photographs up. And especially I want to thank the Howard Hughes Medical Institute for giving me, an evolutionary ecologist with no training in immunology or parasitology, some amount of money to try pursuing a totally different thing for myself and really try and head off in a new direction that I found just profoundly exciting. And with that, I will take some questions.
Here, over here. I'm thinking there's a tie-in between what you're doing and what Ming Wang was doing. And my question relates to your ROS, reactive oxygen species. How long do these animals live with the fibrosis and the ROS? And can you separate those phenomena?
Yeah, so that's a great question. We do not yet see any lifespan penalty with this fibrosis, but it's hard to separate from the parasite itself, which eventually grows to be so big that it lyses the fish. So that's the parasite doing it. We do now have a system where we can induce fibrosis artificially without the tapeworm successfully. And so we're looking at that. I will say the fish typically live about a year in the wild. In the lab, we can get them up to about 4 years. They very rarely live much longer than that in the wild. Their reproduction basically exhausts them. They're univoltine. They reproduce once. They put their all into reproducing in one season, and they just exhaust themselves.